So in photosynthesis, we have two processes, right? What are the two processes? Light dependent reaction and light independent reaction of Calvin cycle. Light dependent reaction takes place in the thylakoid. Calvin cycle takes place in the chronon. So over here, this is the membrane that was found in the thylakoid. So then what process am I talking about here? The light dependent reaction. Is that clear? Okay. So then let's see what happens exactly. So this is a process now. So every process is like a story. So try to pay attention right from the beginning. You don't have to read anything. I'm also not going to refer the resource book, right? Let's just look at the story. What happens? Okay. So now you have light. Light. This light excites this photosynthetic pigment. That excitation or that energy is transferred from this photosynthetic pigment to this photosynthetic pigment. From this photosynthetic pigment to this photosynthetic pigment. From this photosynthetic pigment to this photosynthetic pigment. From this photosynthetic pigment to this pair of chlorophyll A molecules. So now what exactly is this? This is photosystem. This is a photosystem. What is a photosystem? A complex containing of photosynthetic pigments, other molecules, and organic compounds present where? In the thylakoid membrane. So which photosystem is this? This is photosystem 2, or also known as P680. 680 what? 680 nanometers. So that means this photosynthetic pigment, specifically the chlorophyll A, is sensitive to which wavelength of light? 680 nanometers. Right. So what happens? Excite, and that energy is then transferred to this chlorophyll A molecule. Then the electron that was found in the chlorophyll A molecule will go from a lower energy state to a higher energy state. That higher energy state electron is accepted by this primary electron acceptor. Now remember, when we spoke about this before, when chlorophyll loses its electron, what does it form? Chlorophyll plus. Chlorophyll plus. So initially, what was it? A neutral compound. Now it is a positively charged compound. So now this photosystem should work many times, not only once. So now, if this light ray causes it to go from chlorophyll to chlorophyll plus, then it can't work anymore. It has to come back to this chlorophyll. That is where water is important. So now when we talk about the photosynthesis process, we say carbon dioxide plus water gives rise to glucose and oxygen. So now the reactants were what? Carbon dioxide and water. Over here only we use water molecule. What happens is, now since this electron from the chlorophyll is excited and sent to this primary electron acceptor, this chlorophyll will now become what charge? What charge? Positive charge. Now this positive charge has to be neutralized. Is it from this class? No. This plus charge, for it to be neutralized, what does it need? A negative charge. What is the negative charge thing that we know? An electron. This is where water comes into play. Water splits, giving rise to hydrogen, oxygen, and electrons. There is an enzyme present to split, to break down this water to give rise to H plus, oxygen, and electrons. Then the electron that was generated from this reaction can neutralize this positively charged chlorophyll. Then again it is neutral. Then it can work again and continuously this process cannot. So now what can you notice here? Water is broken down into what? H plus, oxygen, and electrons. Oxygen. This is how oxygen is generated in the process of photosynthesis. Is that clear? Okay. So now what is the purpose of this? The purpose of light-dependent reaction is to synthesize ADP and NADPH. So far, we have not talked about NADPH or ADP. Okay, we'll come to that. Now, just as this is occurring, another light ray can come and excite this photosynthetic pigment. Then what happens? That will send the excitation here, here, and then to this chlorophyll A molecule. Then the electron that was present in the chlorophyll A molecule will get excited, and that will move again into the primary electron acceptor in which one? Photosystem 1. What do you call this? 700 nanometers. So this is also known as P700. This is P6A. So again, same problem comes in. Chlorophyll. This chlorophyll A molecule will form a reaction like this. Then this chlorophyll is what charge now? Positive charge. What you think is another water will come here and neutralize this positive charge. But that's not what happens. What happens is the excited electron that was here in the primary electron acceptor of photosystem 2 will go from a high energy level down these molecules that are present in the membrane of the thylakoid. Keep in mind, that is why I have drawn this bilayer to show you that this is present in the membrane of the thylakoid. So the membrane of the thylakoid does not only contain photosystem, but also other molecules present. Okay, PQ, cytochrome complex, and PC. According to your syllabus, they haven't even given the full name. So as long as you remember it like this also, it's enough. Okay, right. So then this excited electron from this primary electron acceptor will go and neutralize this positive charge. From a high energy level, it will go to a low energy level as it is traveling through these transporters, as it is traveling through these components in the thylakoid membrane. That electron will come and neutralize this chlorophyll molecule. Then what happens? It will go back and form a normal chlorophyll. The problem is, or not the problem, the actual function of this is, as this electron is going from a high energy level to a low energy level, it is going through this process or this transport. This is known as an electron transport. So in this electron transport chain, the electron will go from a high energy level to a low energy level. But what is the reason for the electron to go here? It is to neutralize this positive charge. As it is going there, it goes from a high energy level to a low energy level. So when this happens, energy is released from this electron. That released energy is used to synthesize ATP. So exactly how it is synthesizing ATP, we will come back to that in a bit. That is also not necessary for our syllabus, but it's a fun thing to know. Okay, right. Then what happens? Then this electron is excited. That electron is now sent to the primary electron acceptor of photosystem 1. Now this electron that is here will go through another electron transport chain and cause NADP plus that is present to be converted to NADPH. That is why it says NADP plus reductase. What does that mean? It is reducing NADP plus. So what is reduction? Gaining of electrons. So NADP plus will gain H plus and electrons. So when this happens, this NADP plus will become NADPH. Where does it get the energy for that? From this electron which was excited. Okay, so this electron which is excited goes down this electron transport chain and synthesizes NADPH. Is that clear? So this chlorophyll became positive charge, neutralized by this electron. This chlorophyll which became positively charged became neutralized by this water. So far from this light dependent reaction, what are the products that are formed? Oxygen is formed, NADPH is formed and ATP is formed. This ATP and NADPH is the main purpose of this light dependent reaction. Why? Because this NADPH and ATP is used to synthesize glucose in the Calvin cycle. Is that clear? Right. So now I just told you that it synthesizes ATP. Let's see exactly how ATP is synthesized. This is extra information. Right? So as the electron is going from a high energy level to low energy level, it releases energy. That energy is used to pump in hydrogen ions from the stroma. Now, how do you find the stroma here? If I draw a quick diagram of the chloroplast, where is all of this happening? In one of these areas, no? One of these areas. So outside the membrane, what do you find? Stroma. So the H plus from the stroma comes into the thylakoid lumen. That means the inside of the thylakoid. Why does it do that, or how does it do that? Due to the energy that was released when the electron is going from a high energy level to a low energy. So now what does this do? This builds up hydrogen ions inside the thylakoid loop. Hydrogen ions inside the thylakoid loop. There is another protein found in the thylakoid membrane known as ATP synthase. What ATP synthase does is, as the hydrogen from the inside goes outside, it uses the energy of the movement of hydrogen to generate ATP. This is extra, but if you wanted to know extra knowledge, this is how it exactly works. Understood? Light dependent reaction. The last part. So now as the electron is going from a high energy level to a low energy level, it releases energy. Yes, that energy is used to bring in the hydrogen from the stroma, that means outside the thylakoid, into the thylakoid. Okay, then the hydrogen ion concentration inside the thylakoid will increase. Right. There is another transporter present. There is another transporter present in the thylakoid membrane known as ATP synthase. What it does is, it allows these hydrogen ions to move out. Since this area had a high hydrogen concentration, this area had a low hydrogen concentration, hydrogen will now go out. Now remember, I don't think I told you this, you didn't come last week, no? That in a flowing river, if you put that water wheel, as the river is flowing, the water wheel will rotate and that will generate electricity. Or if you take a windmill, as wind is blowing through the windmill will rotate and that generates electricity. Just like that, hydrogen is going from a high concentration to a low concentration. This movement of hydrogen will result in ATP synthesis. Is that clear? Okay, anyway, this is extra information. All you need to know is, as the electron is going from this primary electron acceptor of photosystem 2 to neutralize the chlorophyll A molecule in photosystem 1. This electron transport chain is what is important in ATP synthesis. This electron transport chain is involved in NADPH synthesis. The name of this enzyme is NADP plus reductase. If you say NADPH reductase, it's false. Why? Because you're not reducing NADPH, no? You're reducing NADP plus. So keep that in mind. Okay? Right. So this is linear electron flow. Why do you call it a linear electron flow? See how the electron is moving in like a line. Right? So it's going through this electron transport chain, then up and again electron transport chain. So it's going in like a line. That's why you call it linear electron flow. Okay. However, in this light dependent reaction, there is another part known as cyclic electron flow. In cyclic electron flow, what happens is, in this only photosystem 1 is involved. There are in a certain situation, okay, you will come across this in a certain situation in the C4 pathway, in the C4 plants, we will do that later. This PS1 is capable of exciting its electron right? In the normal pathway, we saw that this electron is going through this electron transport chain and synthesizing what? NADPH, right? In the cyclic electron flow, what happens is it uses another pathway. It uses another pathway and synthesizes ADP. So what happens in this pathway is, see, chlorophyll is excited and the electron from the chlorophyll goes to the primary electron acceptor. Then chlorophyll A molecule becomes positive. Then this primary electron acceptor through another pathway will generate ATP. And that electron is again coming back to what? Chlorophyll. So the electron that was given out by the chlorophyll goes through another pathway and again comes back to the chlorophyll. Then the positive charge will be neutralized. So in this process, you are generating only ADP. Okay, is that clear? Right. So that concludes light dependent reaction. Okay, time to watch the next part of the movie, like Avengers Infinity. You have watched Avengers? Oh, thank God, right. Avengers Infinity War, and now it's Avengers Endgame, because we have reached the end game of photosynthesis. That deserved an applause. Come on. Okay. So now, what happened in light dependent reaction? The entire linear electron flow happened and what was produced? NADPH and ADP was produced. Now we need to use this NADPH and ADP in some manner to generate the product that we require. And in photosynthesis, that is glucose. Right. So now, what happens? You have this carbon dioxide. So again, in the process, in the reaction of photosynthesis, it's carbon dioxide plus water, glucose and oxygen, right? Carbon dioxide over here, water we saw in light dependent reaction. This is where carbon dioxide is used. Carbon dioxide binds with a molecule known as RUBP, ribulose bisphosphate. It is a 5-carbon compound. This carbon dioxide and this RUBP join together to form a 6-carbon intermediate. Why is it known as a 6-carbon intermediate? Because this 6-carbon molecule is not stable. So this 6-carbon molecule will eventually break down into 2 3-carbon molecules. Is that clear? The first stable product of this cycle is 3-phosphoglycerate. Please remember these terms, surely will be asked. 3-phosphoglycerate. Now this carbon dioxide being attached to this RUBP is a reaction. And as every reaction requires an enzyme. The enzyme that is used in this process is known as Rubisco. Rubisco is RUBP carboxylase oxygenase. RUBP carboxylase oxygenase is known as Rubisco. Okay, right. So RUBP carboxylase, that means adding a carbon to RUBP. Remember it like that. So RUBP carboxylase, here you are adding the carbon dioxide to RUBP. Therefore, RUBP carboxylase. So then what does RUBP oxygenase mean? Adding an oxygen. So this enzyme is known as RUBP carboxylase oxygenase. So what does that mean? It can add a carbon or it can add a oxygen. So for example, if there was a lot of oxygen, what will it add? Oxygen. If there is a lot of carbon dioxide, it will add. Okay. That is relevant in the future. So this carbon dioxide is added to this RUBP to form this 6-carbon intermediate using the enzyme Rubisco. So this enzyme Rubisco, where will it be found in? Link your information. Yeah, stroma. Why? Where does the Calvin cycle take place? So where is the Rubisco used? Where is the Rubisco used? In the Calvin cycle. Okay, so then therefore where is it found? You can connect information like that. Okay, right. So then this 6-carbon intermediate will break down into their 3-carbon stable compound. That compound is known as 3-phosphoglycerate. This is known as a 3-phosphoglycerate. So over here, how many phosphoglycerates have I drawn? 6. Okay, right. Why have I drawn that? I will tell you at the end. So now this adding carbon dioxide to this RUBP is known as carbon dioxide fixation. That is the first step in the Calvin cycle. Is that clear? This 3-phosphoglycerate will then be converted to 1,3-bisphosphoglycerate by the addition of AD. It is very easy to remember this. Why? When you study organic chemistry, you will know 3-phosphoglycerate means this 3, the number 3 is showing the position of the phosphate group. Okay, so 1,3-bisphosphoglycerate means it is showing the position of the phosphate group again. One phosphate group will be in carbon number 1, the other phosphate group will be in carbon number. Okay. Over here, the phosphate will most likely be in carbon number 3. So number of phosphates is increasing, right? So ATP can remove its phosphate. You know that no, ATP can become ADP plus inorganic phosphate. So that phosphate is given. So very easy to remember that 3-phosphoglycerate becomes 1,3-bisphosphoglycerate by the addition of ATP. This process requires ATP. Is that clear? Okay. Then you have the reduction phase. What does reduction mean? The specific compound that we are talking about here will undergo reduction. Is that clear? Okay. So what is the compound here now? 1,3-bisphosphoglycerate. Right. This 1,3-bisphosphoglycerate will become glyceraldehyde 3-phosphate. Okay. In this process, you require ATP and NADPH. ATP and NADPH, if I recall correctly. Yeah, right. So reduction means what? That compound is gaining electrons. Yes, right. So this specific compound, let's say 1,3-bisphosphoglycerate, is gaining in hydrogen. Why? Because we learned that when you are giving a hydrogen, you give it as H plus. So since there is a plus going, there should also be a electron or minus going to neutralize. So since this minus is going, we call it a reduction. Okay. So this 1,3-bisphosphoglycerate will become glyceraldehyde 3-phosphate. The phase is the reduction phase. That means this compound is undergoing reduction. If it is undergoing reduction means the H from the NADPH along with electrons are being added to that compound. So when that happens, this 1,3-bisphosphoglycerate becomes glyceraldehyde 3-phosphate. Is that clear? So along with NADPH, ATP is also required because energy is required for everything. Therefore, ATP should be present. This is how you remember. Right? Try to link this information to something that makes sense. So it makes sense. Understood? Okay. So then this 1,3-bisphosphoglycerate will become glyceraldehyde 3-phosphate by the use of ATP and NADPH. Since NADPH is being used here means it is giving a hydrogen to this molecule. So if it is giving a hydrogen to this molecule is undergoing reduction. Therefore, reduction phase. Right. Then, so all these molecules will undergo reduction, which will give rise to 6 glyceraldehyde 3-phosphate molecules. Now I will tell you why I have drawn 6. Okay. So we started with 3 and 3 RUBPs. Okay. So we have a point in the resource book that says the glyceraldehyde 3-phosphate, after it is being given off, it will regenerate RUB. But 3 carbon cannot become 5 carbon. It can't do. But if you have, let's say 1, 2, 3, 4, 5. How many? 5 multiplied by 3 is 15. 3 multiplied by 5 is 50. So over here you can see that this glyceraldehyde 3-phosphate can leave to generate glucose. Why? Because this remaining is involved in regenerating what? Regenerating RUBP. So regeneration of RUBP, guess what you need? ATP. For regeneration of RUBP, you need ATP. Then this process continues. So that is why, see, you are adding 3 carbons. What are you getting at the end? A 3 carbon. So to give rise to a 3 carbon molecule, which is glyceraldehyde 3 phosphate, is a 3 carbon molecule. So to give rise to a 3 carbon molecule, how many times should this cycle take place? 3 times. That is why I have drawn 3 carbon dioxide molecules, because it is difficult to understand with a single one. Is Calvin cycle clear now? That's it for photosynthesis.